Hello, everyone. I'm Delphine Lariviere, and I work at Penn State University and the Galaxy Project US team. And I'm here today to talk about a joint project with VGP, Vertebrate Genomes Project, to implement reference genome assembly in Galaxy. Galaxy is a framework dedicated to biomedical analysis initially, but since extended to several work of study. And it allows access to the latest analysis tool, latest reference data, and full functional workflow functionality. The goal of this framework is to allow big project analysis and allows to handle thousands of samples through long workflow processing. You can also access Jupyter as Studio directly on the datasets for more detailed analysis. And part of the Galaxy project is also to implement training and training structure and gather a large community of users and developers for answering to any need that the community would have. The framework is accessible through diverse public platforms for free. The framework can also be installed on server or laptop, or even for some people on Raspberry Pi. The VGP project is a several phase project and currently in the first one. The first part of the project aims to target several clades of species and sequence over 260 species to begin with. Further species will be sequenced in later parts. So the first part of this project was running on DNA Nexus and Amazon Web Service, which is not free and was accessible mainly for the people from the project. And when people in VGP wanted to develop the second version of the pipeline, we collaborated with them in order to make it free and accessible for everyone and not just the member of the VGP project. So this version 2 in Galaxy resulted in about a dozen workflow that you can see here. Each box is a workflow. The first part is using HiFi PacBio reads in order to build initial contigs. 3 different paths are available for this initial contigging, including a trial mode if Illumina data are available for the parents of the individual sequence with PacBio. One of these workflow is optional depending on the result of the contigging. The contigs can be purged depending on the data available for the project. If CMAP Bionano data are available, scaffolding step can be added in the middle before the final scaffolding using high-C data. And once the scaffolds are built, a couple steps of decontamination for either foreign organism or mitochondrial genome can be done before sending for manual curation. This workflow are available on GitHub, so they can be downloaded and then uploaded to any Galaxy instance if the tools are installed. They are currently in pull request, but they will be eventually available in the IWC, in the Galaxy Workflow Commission, that contain a large number of workflows that have been created and developed for state-of-the-art analysis. These tools are available for free in the US platform or the European platform, .org and .eu, and also in progress of being installed in the Australian platform. So in addition to this free access to the analysis, they have been used for the last 6 months to assemble over 21 genomes using the free resources and for people, both in the VGP project and students who are working with them, the largest one being toad, the eastern narrowmost toad, and also include the wallaby and the Hawaiian crow. In addition to the workflow accessible, we built training material for people who are not experienced in genome assembly or are new to Galaxy or want to know what to do with the data. We have two versions, one long and one short. The long one detailing every tool and the choices we made for the parameters, and the short version teach how to use the workflow as black boxes and get to an assembly simply and quickly. So the longest version takes about 3 hours, the shortest one about 1 hour. And we've been using this training to teach in several opportunities. We have a Galaxy Tapas Day that happened in March 2022, and will happen in spring next year, which is a free online event where we have people from all over the world accessing any training that we want, and we are online to help them and guide them for any problem that they have. With the VGP project, we also have been going to workshop and symposia to collaborate and include people from any horizon in the project, including Galaxy Europe Biohackathon in November 2021, where we included people and what they needed to be included in the workflow. And in May 2022, we participated to the first Africa BP Open Institute for Genomic and Bioinformatics workshop on endemic African species. And we have many other training events planned to include everyone we can from the community. This project has been made possible by the VGP team and the Galaxy team, and everyone in the Galaxy community that maintain this framework and develop tools from everywhere. So thank you for your attention and have a great day.